Ma poi ci mettiamo qua? No, ci mettiamo qua. E poi facciamo. Perfetto. Hi everyone. If you've all taken your places, we can start. Good evening to everybody and welcome to the first lecture of the 2025 edition of Horizons in Medicine and Biology. I am Lorenzo Della Casa and this is my colleague Alessio Morosi, both second-year medical students here at Scuola Superiore Sant'Anna. Before we begin, here's a brief introduction for those unfamiliar with this institution. The Scuola Superiore Sant'Anna is a public university that admits students purely on merit through a highly competitive national selection process. Students at the school have the opportunity to tailor their university studies to their personal interests. They thrive in a dynamic, multidisciplinary environment where individuals from different areas come together to participate in shared courses and projects while living in the same community. These collaborative settings fosters the exchange of ideas and nurtures innovation.
Thanks to this event, Scuola Superiore Sant'Anna is thrilled to welcome distinguished experts in the biomedical field as one of us for a day. The host will join our community, sharing his expertise and journey with students and faculty alike. This unique opportunity to connect with such inspiring figures underscores the school's dedication to creating moments of meaningful interaction and inspiration. Students will gain invaluable insights not only in the latest advancements in medicine, but also in the professional path of leaders in the field. For one day, they become part of our academic family, leaving a lasting impact and sparking new ideas that could shape the future careers of many. Before the start of the lecture, we would like to share with you a short summary of the past edition of Horizon in Medicine Biology through this video. Okay, thanking Professor Balligand for being with us today. I give the floor to Professor Vincenzo Leonetti, the architect of this seminar, who will introduce our guest. Thanks all to you.
Buonasera a tutti, welcome to everybody. And it is a big honor for me to introduce Professor Jean-Luc Balligand from the University of Louvain. And just a few information about his career. He got his medical degree in 1984, and he was assistant professor of medicine in 1995 at Harvard Medical School. And then he came back to Europe at University of Louvain, and actually he is full professor of medicine and pharmacology. Jean-Luc Balligand is a physician, but he's a great scientist, and he got amazing results in order to understand endothelial dysfunction and even some underlying mechanism related to nitric oxide release, or mechanism that can be impaired after long-term exposure to LDL cholesterol. For this and other work, he received several awards, relevant awards, like the Terabel Prize, the Galien Prize, and the Vestager Prize of the Belgian Society of Cardiology. Currently, he's head of the Pole of Pharmacology and Therapeutics and president of the Institute of Experimental Research and Clinique of the Faculty of Medicine at University of Louvain. He's a physician, as he still practices medicine with patients and is still continuing to perform experimental activity, both. The profile of Jean-Luc Balligand is a physician scientist that is actually a rare profile, and he's a member of the Royal Academy of Medicine of Belgium and board member of the Health Competitiveness Cluster of the Wallonian region, BioWin. So it is a great pleasure to invite on the podium Professor Jean-Luc Balligand. Thank you.
Thank you for this very nice introduction, Vincenzo. Actually, when I look at the list of previous speakers on this podium, I'm really humbled and honored to have been invited to give you this lecture. I also met the lady rector of the Sant'Anna, and then was very impressed by all the initiatives that have been and are being taken for the benefit of all these bright students who attend Sant'Anna. I think that this is a unique environment for them, but also an initiative that should be duplicated in many other institutions to really prepare the faculty of the future in medicine, be it experimental or clinical. So thank you very much, Vincenzo, also for the nice introduction on my previous work. And today I will try to recapitulate many years of research, not only done by me, but most importantly by young people like many of you in the audience, who certainly deserve all the credit for the work that I will show. And try to convey to you a glimpse of what you go through when you try to verify a hypothesis that is out of the common thinking. So, thank you for putting this in presentation mode, if you will. Yes, thank you very much. So,
Now, right now? Okay. Maybe the other.
Doesn't seem to work. Oh,
Okay.
yes.
It's the other one.
So this is what we have to deal with, which is the transformation from a normal heart to a diseased heart. It's an enormous problem in cardiology. There are many circumstances that drive these changes that we can call adverse remodeling. And this entails different aspects, changes in the hypertrophy of cardiac myocytes, but also changes in perfusion. I mean, there's a renewed interest for microcirculation, and that drive ischemic disease, even subclinical ischemic disease that participates in this remodeling. And also another important aspect is fibrosis. If you would please hide this panel, this would be excellent. Thank you. And you know, there's a renewed interest for cardiac metabolism, and certainly the adverse remodeling also entails a number of important changes in the way the heart uses metabolic substrates and how it performs its metabolism. So I think everybody in medical school knows what excitation-contraction coupling is. And what you see here are classical elements of it with, you know, the plasma membrane diving into T-tubules. And then the first element, which is the voltage-sensitive calcium channel that drives the entry of calcium into the fuzzy space, then activating calcium-induced calcium release that, you know, promotes the contraction of the contractile apparatus. And then this is followed by reuptake of calcium into the sarcoplasmic reticulum. That is also an important aspect of relaxation. And it's known to everybody that all these aspects of excitation-contraction coupling are modulated by catecholamines, mainly acting on adrenergic receptor, and more specifically in cardiac myocytes on beta-1 and to a less extent to beta-2 adrenergic receptors. On the acute stimulation, this is adaptive. It's part of the fight or flight reaction in face of a danger. But over the years, it became understood that chronic stimulation by this and other neurohormones is deleterious because it drives excessive calcium release. And this is accompanied with changes in the expression and function of some of the key proteins that drive this excitation-contraction coupling, resulting in increasing in diastolic calcium and then promoting impaired relaxation, but also apoptosis and ultimately myocyte death. So most of the therapeutic approaches now have been trying to accommodate these deleterious effects by interrupting signaling. And this was the basis of the development of beta-1 and also beta-1/2 blockers, but more specifically beta-1 specific blockers to try and interrupt these chronic deleterious effects. But over the years, it also became apparent that catechols may also act on other receptors that somehow would be protective. You know, if you look at the recent literature, the alpha-1 adrenergic receptor has been postulated to promote some protective effects. And it's not unusual to see in physiology that any stimulus is also accompanied with a counter-stimulus or a negative feedback that helps to control homeostasis. And so we wanted to test this in a very simple setting. And so together with Chantal Gauthier in Nantes at the time, we went directly for human biopsy specimen. So this is a tracing of an increase in peak tension generated by a piece of human myocardium in an organ bath, a very old-fashioned experiment. And when you expose this biopsy to increasing concentrations of norepinephrine, you know, a prototypical catecholamine, then as expected, based on what I explained to you, you would see an increase in the force that is developed, because this is the translation of the positive inotropic effect on excitation-contraction coupling. So that's expected. But then we did another experiment, which was to repeat the experiment of exposure of these biopsies to norepinephrine, but in the presence of a blocker of both beta-1 and beta-2. So there, the catechol can no longer activate 1 and 2, and we would have expected an annihilation, a flat curve. But in fact, this is not flat at all. It goes down. So it's a totally opposite reaction to the catechol that involves a receptor that is neither 1 nor 2, because we're in presence of saturating concentration of beta-1/2 blocker. And so this suggested that there was another adrenoreceptor involved. And not shown here, we repeated this with isoproterenol, which is a synthetic catecholamine that is specific for beta. And so this drove us towards the hypothesis that a third isotype of beta-adrenoreceptor was involved. This was the very first indication for us that beta-3 might be involved. And then when I teamed up with Chantal Gauthier, I was just coming back from the States. And in the States, I had actually started experiments to verify if cardiac myocytes would express some isoforms of nitric oxide synthase that were being discovered at the time. And these are some of the experiments. I won't go through details, but basically, my initial work showed that cardiac myocytes express all 3 isoforms of nitric oxide synthase that produces nitric oxide. And at the time I had in mind, nobody knew what this nitric oxide would do. But I came from the vascular biology. I had done my initial research in the lab of vascular pharmacology, where nitric oxide opposes the contraction of the vascular smooth muscle. And I reasoned, why wouldn't it be the same in the heart? Why wouldn't nitric oxide in the heart also oppose the inotropic effect of catecholamines? And so with that in mind, I went back to Chantal's lab and I suggested to her, why don't you repeat the experiment here, but in the presence of an inhibitor of nitric oxide synthase, because I have proof that these human biopsies express nitric oxide synthase. And let's see what it does. So we added L-name at the time, which is a NOS inhibitor. And what you see in black are actually the quantification of this decrease in peak contraction. But when you put a NOS inhibitor in white, you see that it's clearly attenuated. So the lesson from this is that catechols activate not only 1 and 2 to produce a positive inotropic effect, but at the same time activate a countervailing mechanism that attenuates the inotropic force through nitric oxide synthase. I can summarize this in this cartoon that is now more complete. So you have beta-1 that activates CC coupling. We know there is N-NOS and E-NOS, and the third isotype activates these NOS. We had the first functional hint of this with the L-name experiment that would oppose the effect of beta-1. So it's the negative feedback, if you will. It acts like a smart beta-1 blocker. But is it just a smart beta-1 blocker? Would it do something else to the myocardium, especially having in mind that adverse remodeling is driven by excessive beta-1 stimulation? So would the beta-3 oppose these deleterious effects? That was the natural question. But before we embark on that, I need to give you a little bit more background on beta-3. It had actually been identified as a metabolic receptor. So Donny Strasberg, who's Belgian, by the way, he used to teach at the University of Brussels and then moved to the United States. He was in Cochin in Paris when he did this work. He basically screened a human adipocyte DNA library and identified the gene coding this third isotype. So what do we know about this isotype? Well, this is a representation of the primary sequence. It's a GPCR. So as a GPCR, like all the other GPCR, especially in the beta-adrenergic family, it has 7 transmembrane domains. It has a N-terminus that is glycosylated, as some intracellular loops, and the C-terminus. And what you see in black are the residues that are common to the beta isoforms or isotypes. What is in white are the residues that are specific to the beta-3 and only on the beta-3. And among the residues in the C-terminal tail are the consensus sequences for the phosphorylation by protein kinase A and BARC, or GRK2, G-coupled receptor kinase, that are key for the phosphorylation of the receptor that then attracts beta-arrestin for the homologous desensitization. This is a well-known mechanism of desensitization of the GPCR. When beta-arrestin binds, then the receptor is addressed to the clathrin-coated pits and is internalized. Well, these consensus sequences do not exist on the beta-3. And this confers a resistance to homologous desensitization and internalization. So this receptor stays coupled even in the face of high concentrations of agonists. And moreover, when we put our hand on a trustworthy antibody, we looked at the tissue expression of this beta-3. So it's well known to be expressed in adipose tissue. But with my associate Chantal Ducy, we found it in human endothelial cells. We confirmed it's present on human cardiac myocytes. And there are some indications for expression in the pulmonary vasculature, even though this is more controversial. But that's not the end of what you should know. The other thing is that using this antibody for immunohistochemistry, we found that it's expressed in normal hearts. So let me drive you through this. These are the negative controls. This is a control heart. It's basically a non-diseased heart that's improper for transplantation. And these are hearts from dilated and ischemic cardiomyopathy. You clearly see there's an increased staining. We confirmed this quantitatively at the transcript and protein level. And so the conclusion is that this receptor is upregulated in diseased hearts. It's the opposite of what is known for beta-1 and beta-2. With cardiac disease, beta-1 and 2 are downregulated. The third isotype does exactly the opposite. And remember, it stays coupled. It's not desensitized. So this makes it a perfect target for activation in a diseased heart because it would still convey signaling. So we thought this was worth pursuing, provided we could demonstrate that this receptor would do better or more things than just blocking beta-1 signal. And indeed, over the years, we confirmed that this receptor is coupled through G-alpha-I to the NOS and produces nitric oxide and activates the production of cyclic GMP. Cyclic GMP has many pleiotropic roles. We'll come back to that, both on cardiac remodeling and other aspects of myocardial biology. And again, we showed that all these effects consistently oppose the deleterious effect of beta-1. But does this signaling protect from adverse remodeling, which is the initial problem that I addressed at the start of the lecture? So there's no better approach for that than a reductionist model with mice. Why mice? Because with mice, we can obviously collect easily tissue, do longitudinal study, and study all the aspects that I mentioned before. And I will address all these aspects: hypertrophy, fibrosis, altered metabolism, and we'll ask the question, does the beta-3 influence the progression of these adverse remodeling? And as I said, the best reductionist approach preclinical is to generate a mouse. You should know that the mouse heart has naturally very, very low levels of expression of the beta-3 isotype, at least in the normal state. So we reasoned that we needed to express it exogenously. And so we used a transgenic mouse model with the gene coding for the human beta-3 isotype driven by a cardiac-specific promoter, alpha-MHC. And we use all animals in the heterozygote state. Why? Because we wanted low levels of expression of the human beta-3. And we even verified that this low level was quite comparable to what we found in human biopsies. You see that grossly the amount of beta-3 expressed in our transgenics parallels what is seen in human hearts. And then we exposed these mice to a number of interventions, including transaortic constriction, with a moderate velocity gradient. And again, we didn't want to put them in failure. We wanted to just produce this remodeling without driving them to failure, because we wanted to see whether we could interrupt this process without the confounding effects of a full heart failure phenotype. So that's what we did. And all the rest that I would show you is due to the work of a few brilliant PhD students and postdocs in the lab, most prominently Lauriane Michel, who should be giving this talk, but has been really instrumental in generating this data, as well as all the team. So what I will show you is first the data on cardiac morphology between the transgenics and the wild type, littermate, submitted to transaortic constriction. So for all the rest of the slides, yellow is transgenic, so with beta-3. Wild type with no beta-3 or very little is in black. First thing, this is total heart weight to tibial length. So after TAC, you have a significant attenuation of hypertrophy. This is confirmed by echo on LV mass index. And when you look at the transverse section of myocyte histologically, you see that the wild type develops myocyte hypertrophy, and this is significantly attenuated when you express the human beta-3. Importantly, this attenuation of the remodeling is not at the cost of loss of function. In fact, the wild type under TAC seems to lose a little bit of left ventricular function. This is a gross index. It's not the most accurate, but we have other measurements. It's ejection fraction. But it's better preserved, even in the transgenics. So less remodeling without compromising cardiac function. Now, I mentioned to you that remodeling hearts undergo changes in metabolism, and usually the normal heart has an enormous substrate flexibility, can use glucose, can use fats, mostly relying on fat on the normal state. But under stress, usually there's a shift towards glucose utilization. And in adverse circumstances, usually this glucose doesn't go to the way through oxidation, but there is aerobic glycolysis that produces lactate with, adverse effects in terms of cardiomyocyte pH and survival. So having these metabolic changes in mind that we expected, we looked at first glucose uptake, and this is an experiment in vivo in our mice with F18-FDG uptake. And as expected, 1 week after transaortic constriction, you see that both the wild type and the transgenic increase their uptake of glucose. So that's expected from a stressed heart. But then we did a longitudinal study at 3, 6, 9 weeks, and you see that progressively the wild type decrease slightly their capacity to take up glucose while it's maintained or better maintained in the transgenics. So it's even more prominent if you isolate the cardiomyocytes from these mice, the same mice, and submit them to insulin and then measure the uptake of tritiated glucose. This is at basal state. Then at 3 weeks, you see that there is some insulin resistance. They decrease their capacity to uptake glucose. But at 9 weeks, even though the wild type have completely lost their insulin sensitivity, this is restored in the transgenics. So beta-3 expression somehow helps the heart to take up more glucose. Now, we know that the uptake of glucose uses some transporters. And so we looked at GLUT1, which is the insulin-insensitive transporter, no change. But GLUT4 is lost, or at least decreased in the wild type, but preserved in the transgenics. We confirmed this at the protein level and also at the membrane translocation. So there's clearly a preservation of GLUT4 at the membrane of transgenic mice. Now, you'll ask me, what about the other substrate? What about fat? Well, absolutely no change. We went to great expenses and efforts to develop an in vivo tracer for fat uptake, but it didn't show any significant change. We did this at the myocyte level with labeled palmitate, no change, and no change in the transporter CD36. So it seems to be restricted to the uptake of glucose. What happens to this glucose? Then we went through untargeted metabolomics to try and see whether we could quantitatively measure intermediates of the glycolysis pathway. And we were surprised to see that when we compare the wild type, undertap, and the transgenics, there was a relative decrease in the abundance of intermediates of the upper glycolysis pathway, but a relative enrichment in substrates or intermediates of the lower glycolysis pathway. So this suggested to us that if this is decreased, it means that the glucose is derived towards another pathway. And so we had to examine all of them. So we started with the exosamine pathway and looked at UDP-GLICNAC and UDP-glucose, no change at all. So no evidence for an increase in oglycination. And then we looked at glycogenolysis. And in fact, we didn't see a derivation towards the synthesis of more glycogen. On the contrary, we found that beta-3 were degrading or using diglycogen, and we saw an increase in the glycogen enzymes. So if anything, this pathway is feeding even more glucose into the glycolysis. So next, we then reasoned that if it's not the exosamine, not the glycogen synthesis, it had to be through another pathway, and the obvious candidate was the pentose phosphate pathway. And so this is a representation of the pentose phosphate pathway. And these are the results of a flux analysis with a stably labeled substrate that you see here in a beating heart in a Langendorf mode. And then we analyzed all the intermediates of the pentose phosphate pathway. The quantitative representation is here, but to simplify things, those that are in red are significantly enriched. And so all this was in line with an increased flux of metabolites through the pentose phosphate pathway. And interestingly, UDP-GLICNAC again was not enriched significantly, showing that there's no derivation to the exosamine. But this suggested or demonstrated that there was an increased flux in beating conditions in hearts that express the beta-3 receptor. We also had an upregulation of key enzymes that are in the oxidative part of the pentose phosphate pathway that produce an ADPH, as you see here, as well as other enzymes such as the T-aldo, a little lower in the pathway. As a consequence of the upregulation of enzymes in the oxidative part, more NADPH would be produced that is well known to serve in part in reducing glutathione that serves as an antioxidant. And so we first looked at the generation of NADPH. It was increased, NADP plus was unchanged, and so the ratio of NADP plus to NADPH was significantly down in the transgenics, meaning that this would be expected to be translated with more reduced glutathione, which is the case. You see that it's down in the wild type, but preserved in the transgenics, with an increased ratio of reduced to oxidized glutathione. And we even found an upregulation in the beta-3 of the key enzyme that drives this reduction of glutathione. All this should be translated into more antioxidative capacity. And as you all know, oxidative stress is a key driver of remodeling, be it at the myocyte level, but also in fibroblasts and other cell types. And so we were keen to measure oxidative stress directly in the cardiac myocytes of our mice that had undergone TAC after 9 weeks. And what you see here is the amount of DCFDA fluorescence under H2O2 in the transgenics that is significantly lower. So showing that there is an increased antioxidant capacity in the transgenics. And another marker of oxidant stress was also significantly decreased under beta-3 expression. So clearly this beta-3 drives an increase in flux through the PPP, and the oxidative part of the PPP underlies an increased detoxifying capacity towards reactive oxygen species. And all this would be expected to be protective. Now, an important other aspect is the possible production of purines and pyrimidines in these hearts, because some of the intermediates of the PPP drive the synthesis of purines that is important for DNA synthesis, and some have postulated that hypertrophy is helped by increased synthesis of nucleotides that drive increased transcriptions of the genes that participate in remodeling. But in fact, we didn't find any significant change there. So what happens then to all these intermediates of the pentose phosphate pathway? As I showed you before, remember there was a relative enrichment in lower glycolysis intermediates in our untargeted metabolomic approach. And so we looked at the possibility that these lower glycolysis intermediates would actually be driven towards glucose oxidation in mitochondria. And indeed, we found a significant increase in pyruvate, but no change in lactate. Remember, I mentioned to you that in most pathologic circumstances, the heart relies on glycolysis, but produces more lactate. That is deleterious. And in fact, this is not the case under beta-3 expression. On the contrary, it seems that this glucose is oxidized through pyruvate. And so we needed to confirm this, and we first did a global oxygen consumption, and we found that the expression of the beta-3 would drive an increased consumption of oxygen. We confirmed this in isolated cardiac myocytes, adult cardiac myocytes in the CORS analyzer. And this is the quantification of the maximal respiration. And you see that even though it's slightly decreased in the wild type, it's significantly increased in the transgenics. So, and on these CORS analyzers, we feed the cardiac cells exclusively with glucose. So we have pretty good evidence that this glucose is oxidized. How could this be sustained over 9 weeks? Remember, this is a chronic adaptation, so it needs to be accompanied with some changes also in the mitochondrial capacity. And indeed, we found that expression of the beta-3, I can't go back. Yes, I can go back. Is accompanied with an increase in the expression of some key factors for mitochondrial biogenesis, at least at the transcript level, but we have additional measurements of mitochondrial density, supporting the hypothesis that this increase in maximal mitochondrial respiration is supported by a beta-3 mediated increase in biogenesis and fusion of mitochondria. So, as I said, to be protective, and I did show you that the myocardium was protected from adverse remodeling, this reaction to beta-3 expression needs to be sustained over time. And so this probably entails change in the expression of key genes that would drive this metabolic adaptation. So we did an unbiased transcriptomic analysis on our hearts, and the PCA shows you that we can clearly separate the wild type from the transgenics. And this volcano plot shows a number of genes that are either up or down regulated. And when we do a pathway enrichment or an over-representation analysis by processing our transcriptomic analysis with databases that select for putative transcription factors that drive these changes, then we ranked putative candidates as transcription factors. And interestingly, among the top 5, we found this one, NFE2, which stands for NRF2. And NRF2 is of particular interest to us because when you look at the classical transcription program that is driven by binding of NRF2 to its antioxidant responsive element, we find that all the genes that we found overexpressed in the beta-3 transgenics are among the classical targets for NRF2. And this would drive exactly the kind of phenotype that we would expect, an increase in the oxidative part of pentose phosphate, an increase in glucose uptake, mitochondrial biogenesis. And so all this was compatible and in line with the phenotype that we had. So we went to the hypothesis that probably this expression of the beta-3 would mediate its effect through NRF2. But we needed to prove that. So first of all, we looked at NRF2 expression itself. And indeed, we found a significant upregulation of this transcription factor in the transgenics under TAP. But then we wanted to look at function. And so for that, it's difficult to do it in animals, even though we have some experiments in vivo, but we resorted to an even more reductionist model where we used cultured cardiac myocytes in which we express the human beta-3 through an adenoviral expression. So these ADRB3 AAV infected myocytes were then submitted to a classic pro-hypertrophic agent, phenylephrine, which is an alpha-1 adrenergic agonist. And as you can see, as expected, the control that do not drive beta-3, but just the GFP, develop hypertrophy in the phenylephrine. But if you express the beta-3, then you have a significant attenuation of the hypertrophy. So we reproduce in this culture system exactly the phenotype that we had in our beta-3 transgenics. So that's a good start. And the next experiment was to look at NRF2. So NRF2 is a transcription factor that moves from the cytosol and goes to the nucleus to drive transcription. So we can follow this by immunocytochemistry, and that's what's illustrated here on the left. Under phenylephrine and in the presence of expression of the beta-3 with the adenovirus, we see a concentration of the NRF2 transcription factor in nuclei, whereas it stays in the cytosol when there is no beta-3. So beta-3 promotes the recruitment of NRF2, and we can quantify this by calculating the amount of NRF2 that's nuclear versus cytosolic. And we find that it's clearly enriched in the nucleus. And we even did a functional assay where we looked at transcriptional activity of NRF2, which is clearly driven up under expression of the beta-3. So that was a good start. It doesn't mean that it's causal. If you want to prove causality, you need to show that your beta-3 effect is lost when there is no NRF2. And it's not shown here, but we did these experiments both with an siRNA approach and also with a putatively specific pharmacologic inhibitor of NRF2, and we found that if you downregulate NRF2 or if you inhibit it, then the effect of beta-3 is lost. But for simplicity, I don't show this. But I want to come back to NO. Where is nitric oxide in the picture? You remember that I showed you that beta-3 is coupled to nitric oxide? And so we wanted to see whether nitric oxide had anything to do with the phenotype that we had. So again, we used the same reductive model. And again, those that express the beta-3 are here. And what you're looking at here is the amount of nuclear NRF2. I showed you that it was enriched in the beta-3 expressing cells. But if you do the same experiment in the presence of a NOS inhibitor, L-name, the same I used years ago with the biopsies, then you abrogate this increase. So somehow beta-3 drives nuclear translocation of NRF2 through a NOS-dependent mechanism. And the same with myocyte area or another marker of hypertrophy. You see that even though in absence of L-name, you have a clear attenuation of the myocyte area in the beta-3 expressing. If you inhibit NOS, then oops, you recover hypertrophy. So NOS is a key player in the picture. But how? Well, first of all, we needed to verify this in vivo. So we took our mice again and treated them with L-name in the drinking water for weeks. And even though in the previous slide I showed you that the transgenics would have less hypertrophy, here the difference is abrogated. So if you take NO out of the gain, you lose the protection by beta-3. And this is also true for another index. Moreover, the change in mitochondrial respiration that I showed you before is also NOS sensitive. If you inhibit NOS, then no difference at all between transgenics and wild type. And this is shown again here. So how does NO play into the picture? Well, you need to know that NRF2 is a transcription factor that's kept into the cytosol through an allosteric inhibition with a protein partner that's called KEAP1. And looking at the sequence, the primary sequence of KEAP1, we found that there were at least 3 cysteine residues that are amenable to post-translational modification, including by S-nitrization. You know that thiol residues, SH, are very amenable to covalent modification by nitrization. And we reason that this nitrization may interrupt the inhibitory interaction with NRF2, thereby releasing its inhibition and allowing its translocation to the nucleus. So the first thing to do was to verify whether there was a nitrization of KEAP1. And this is the experiment. It took us enormous effort, not me, but Florian and my team, because this S-nitrization is very sensitive to reducing environment, to light, to time. So it's very labile ex vivo. But with a resin capture approach, we could find, this is the positive control with SNAP, which is an NO donor, that KEAP1 can be nitrizated. And more importantly, we found that if you have a GFP in the presence of phenylephrine, there is very little amount of S-nitrization. But if you have the beta-3 here in the presence of phenylephrine, and even in the absence, you see that there is a significant S-nitrization that is completely abrogated if you put the NOS inhibitor. So this is from extracts of our cardiac myocytes. So it means that beta-3 drives in S-nitrization of KEAP1 that is sensitive to NOS inhibition. This is the quantification of the S-nitrization experiment. And so we come with this picture, which is that beta-3 expressed in cardiac myocytes is coupled to nitric oxide synthase, as has been amply demonstrated by us and others before. This produces nitric oxide. NO then S-nitrosates keep one, releases the inhibitory interaction from NRF2, and NRF2 then is driven to the nucleus. We've quantitatively demonstrated this. And then this NRF2 drives the transcription of a number of genes that we identified through unbiased approach initially, but that are key targets of NRF2. And these promote glucose uptake, but also the oxidative part of the pentose phosphate pathway to generate more NADPH and more reduced glutathione, thereby an antioxidant response. And all these metabolic changes are sustained in time thanks to a transcription-mediated increase in biogenesis, mitochondrial biogenesis and function that drives mitochondrial respiration. And this would, through glucose oxidation, maintain the energetic efficiency of the heart in the face of stress, and probably explain why, despite the decrease in hypertrophy, we maintain left ventricular function. So for those who like text as opposed to images, this is a summary of this first part. So upon prolonged hemodynamic stress, beta-3 mediates an increased glucose uptake, particularly by the recovery of the insulin-mediated glucose uptake through GLUT4. This increase fuels the pentose phosphate pathway, not the L pathways, with accrued NADPH levels, which we measured, associated with a reduction in oxidative stress. And I'll expand more on this, on the functional correlate to that. This is associated with an increased mitochondrial oxidation of lower glycolipid intermediates, supported by increased mitochondrial biogenesis through the NRF2. And this metabolic rewiring is driven by NO-dependent NRF2 activation mediated by S-nitrosation of KEAP1. So that's what we propose based on the data that we acquire. Now, I come back to the oxidant stress, because this is part of the pathophysiology of stressed hearts that are exposed to neurohormones, including angiotensin-1, the alpha-1 adrenergic receptor and beta-1 adrenergic receptor activated by catechols. And this oxidant stress is well known to inactivate the NOS cyclic G pathway through NOS uncoupling, NO inactivation, SGC oxidation. But the antioxidant effect of beta-3, particularly through the oxidative phase of the PPP, would oppose this oxidative stress and maintain the coupling of the beta-3 to these downstream targets. And again, independently, another postdoc in the lab, Nerea Hermida, confirmed that under beta-3 expression and in stressed myocytes, there is a significant decrease in reactive oxygen species, as I showed you before. Now, what would the cyclic GMP drive in addition to the metabolic rewiring that I showed you? Well, cyclic G is known to activate protein kinase G. This kinase has a number of known targets. It's known to inhibit hypertrophy through a number of mechanisms that I will not detail here. We published this a number of years ago, but also phosphorylates troponin I to decrease myofilament calcium sensitivity, which would help diastolic relaxation, but also through the phosphorylation of titin, as shown by others. So in essence, the preservation of this NO cyclic G pathway through the antioxidant effect of beta-3 would help maintain these beneficial effects on remodeling and diastolic relaxation. And indeed, another postdoc in the lab, Charlotte Farah, did examine the phosphorylation of a number of these key players, myosin binding protein C, but also troponin I, in presence of the expression of the beta-3, human beta-3 in cardiac myocytes under stimulation with beta-3 specific agonists, and she did find a significant increase in the phosphorylation of these key proteins. So This seems to be confirmed experimentally. Now, this drives me towards more translation, because you know that this reactive oxygen species, as I mentioned, has a tendency to uncouple the receptor from these downstream effects. And in fact, this is probably because of this reactive oxygen species that some of the previous approaches on cyclic GMP modulation have failed. You know that PD5 and even PD9 inhibitors are not very promising in terms of either promoting diastolic relaxation or inhibiting myocardial remodeling. And this is perhaps because if you want to have an effect of PD5 inhibition or PD9 inhibition, you need to have enough cyclic G produced in the first place. But if the upstream pathway is inhibited or degraded by reactive oxygen species, there's little room for improvement by these PD5 inhibitors. And this is true also for ARNI, so the angiotensin receptor inhibitors and neprilysin inhibitor, even though they have proven clinical efficacy, their efficacy is probably limited because of the little pool of cyclic GMP on which they can act. And other approaches such as HST stimulator and activators have also been rather disappointing, but perhaps through other mechanisms, including on changes in blood pressure. And likewise for nitrite and nitrate, because of all this potential degradation by reactive oxygen species. So adding a beta-3 agonist probably would restore or at least potentiate the effect of many of these treatments by preserving the pathway and the pool of cyclic GMP. This needs to be verified clinically, and I'll come back to that. Now, I just want to also give you some words on fibrosis, because this is the last aspect that I haven't touched on. And again, ROS matter for fibrosis. This is a study that we performed a few years ago where we looked at the collagen volume fraction in human biopsies and correlated to the amount of reactive oxygen species measured by fluorescence. And we do find the linear relationship. So reactive oxygen species probably are a main driver of collagen production and fibrosis. And so again, in our mouse model, we do find that angiotensin II infusion by minipump produces fibrosis, but in the presence of beta-3 expression, this is totally abrogated. And likewise, in our model of transaortic constriction for 9 weeks, you see that there's significant fibrosis in the wild type. It's virtually abrogated, or at least significantly diminished in the transgenics. So this antioxidant effect of the beta-3 really probably matters for the protection against not only hypertrophy, metabolism, but also fibrosis. And again, this is NO dependent because we went to the length of crossing our beta-3 transgenics with mice deficient in one of the isoform of nitric oxide synthase that is coupled to beta-3, the NOS1 in this case. And we found that the protection against fibrosis in collagen volume fraction that we find in the simple transgenics is totally lost when we cross them with the NOS1 knockouts. So NO matters for all these effects of beta-3 in terms of myocardial remodeling, including metabolism through NRF2. So I concentrated on the myocardium, but as I mentioned initially, the beta-3 receptor was initially known and discovered as a metabolic receptor in the adipocyte. So more recently, we reasoned, well, does beta-3 activation in the adipose tissue also matters for cardiac protection? And this is the work done by one of my PhD students, Lorena Cascarano, another Italian. And actually, it's being presented as we speak at the Société Française de Cardiologie in Paris. And so I took her slides, and again, she should be presenting this, but she is presenting this, but in a different environment. But let me just walk you through. So we know that white and brown adipocytes express the beta-3 receptor. And there is some evidence, at least in rodents, that the transition of white adipocytes through different states includes an intermediate state, which is called beige adipocytes, that also express beta-3, and that have an intermediate phenotype between white and brown. These adipocytes obviously increase in amount and mass in obesity, which is, highly prevalent and is also a prequel to cardiovascular diseases, especially cardiometabolic diseases. And this obesity also drives cardiac remodeling. This probably entails a crosstalk between these different types of adipocytes and the heart. But since there's previous evidence that beta-3 activation, at least in rodents, promotes beta-3 beiging or brown adipocytes, and that this is known to protect against not only atherosclerosis, but also against skeletal muscle disease, we reasoned, could there be a similar influence on the heart muscle? So the hypothesis that Lorena has been pursuing is to see whether systemic activation of beta-3 in adipocytes, does it produce perhaps beiging, and would some paracrine mechanism or remote crosstalk protect the heart from adverse remodeling under the obesogenic stimulus? So to do this, she developed a model, again, resulting to mice. She spent a great amount of time to select the right background of the mice. But by doing a thorough comparison with different strains, we found that the C57 Black 6J are the best model to test our hypothesis. And these were submitted to two different types of diet, a CHO clone control diet or a high-fat, high-sucrose diet, which is a recapitulation, if you will, of the Western type diet in humans. And under these two obesogenic diets, this obesogenic diet and the control diet, half of the mice were treated with a beta-3 agonist, the one I mentioned before, the CL316243, to activate beta-3 receptor systemically. So I remind you that the heart of these mice doesn't express significant amount of beta-3. So there we are looking specifically at systemic remote signaling, not directly on the heart. And so these infusion, we decided to infuse this beta-3 agonist both initially at the start of the diet and then to let the mice off the drug. They would again regain weight. And then we decided to implant a second mini pump to see whether any of the phenotype was reversible under beta-3 stimulation. So here are the initial results. So this is the difference initially between CHO diet and high-fat sucrose diet. You see that the high-fat sucrose gain weight more rapidly. These are weeks. This is the weight. And if you submit them to an infusion of the beta-3 agonist, then you look at the other two curves, both under CHO and high-fat sucrose, and you see that the high-fat sucrose then do not gain weight as quickly. If you interrupt the infusion, then the high-fat sucrose regain weight, as you can see here, and the two curves merge again. Now, at this point, if you do a systemic metabolic analysis, then you can look at glucose tolerance, and you see that both these obese mice develop glucose intolerance. That is, they do not correct their glycemia under glucose overload. But when you infuse CL at the end of the obesogenic period, then again, during the second CL infusion, even though the untreated mice keep their insulin resistance, the other treated mice totally regain insulin sensitivity and correct their glucose. So there is a metabolic correction with beta-3 infusion in obese mice. That's illustrated here in the green curve. What happens to the heart then? This is one of the indexes. It's measured longitudinally by echo. So this is just the relative wall thickness, but we have all other indices as well. And you see that the high-fat sucrose diet untreated develop at least this index of remodeling of the left ventricle, but this is totally corrected by CL infusion at the end of the obesogenic diet. And if we look at the transverse area of these myocytes, and this is again done by another Italian who's in the room, who collaborated with us. Alberto measured this, and we did find indeed that the obesogenic diet in control did produce a significant increase in myocyte transverse area, but this was corrected again by CL infusion. And likewise for fibrosis, a significant increase in fibrosis in the non-treated obesogenic diet submitted mice, but a correction with the beta-3 receptor agonist. So this would, it's not complete, this is work in progress, but it seems that systemic activation of this beta-3, we're not talking about the heart directly, but systemic activation attenuates the remodeling induced by the obesogenic diet. We think that there may be paracrine communication between these beta-3 stimulated adipocytes and the heart. We obviously need to identify which mediators are involved, but we have at least a hint because we isolated extracellular vesicles, EVs, from the explanted adipose tissue, both visceral and subcutaneous, of these mice under obesogenic diet. And we used these EVs to superfuse them on cultured myocytes that were treated with phenylephrine. And so this is the, again, the control. So these are without superfusion. So phenylephrine induces a significant hypertrophy, as you can see here. And what you see here is the hypertrophic response under phenylephrine in myocytes that have been superfused with extracellular vesicles from mice that were not treated with the beta-3 agonist. But in contrast, this is the myocytes treated with phenylephrine, superfused with extravesicles coming from the fat of mice that had been treated with the beta-3 agonist. And we suspect that there is some significant beiging with these mice. Actually, we have a number of indices that prove this. And this is the quantitative representation where you see the classic remodeling of myocytes under phenylephrine and the attenuation of this remodeling of myocytes treated with EVs coming from beta-3 treated mice, systemic. So, as I said, it's work in progress. We need to identify exactly which mediators may be contained within these EVs. We need to do metabolomics. We need to do proteomics. We need to look at microRNA. And most importantly, we need to be sure that this is due to beta-3 activation, because we just have pharmacologic suspicion now, but we want to be absolutely sure. And to do that, we're using again, a genetic model where we cross mice that have the beta-3 gene fluxed with mice that drive a Cre recombinase under an adipocyte-specific promoter. So to obtain mice with an adipocyte-specific deletion of the beta-3. And these are being grown now and submitted to the same obesogenic diet with and without the CL. And this will give us the definitive proof that adipocyte beta-3 truly is involved in the remodeling phenomenon that I showed you. If we find significant mediators, then obviously this would perhaps open perspectives for manipulating these mediators pharmacologically or replacing them with drugs that may reproduce the cardiomyocyte protection, but this is for the future. I think I've talked too much. And so I'll finish with some translational aspects, because I promised to you that we would go to translation. All this is nice, but it's cells, it's mice. What about humans? I take your point that we use the human beta-3 construct and human beta-3 receptor, but in a mouse context. So we want to be sure that this is true in humans. Well, there are some hints in terms of left ventricular function and heart failure. My colleague Henning Bundgaard in Denmark actually used the only agonist available for clinical use, which is called mirabegron, a reasonably specific beta-3 adrenoreceptor agonist. And he used this in patients with heart failure. And he looked at the possibility that this beta-3 would recapitulate exactly what we found in the mice, that is an activation of cyclic GMP, and that the cyclic GMP would drive a protection in terms of not only remodeling, but also left ventricular function. And indeed, in this pilot trial, he had a positive outcome with, interestingly, a mechanistic interpretation that would be in line with an antioxidant effect of stimulation of beta-3. So this was encouraging. And before Henning published his study, we had already thought about translational aspects. And given all the data that I showed you on the promising protection by beta-3, I obtained a grant to drive a collaborative work, a clinical study this time, with 10 different recruiting centers. It's called the beta-3 LVH study, where we recruited patients in so-called stage B. And according to the consensus classification, these are patients who already have some structural remodeling of the heart, mostly hypertrophy, but in absence of clinically patterned signs of heart failure. Why did we choose this? Because we wanted again to see whether treatment with a beta-3 agonist in these patients would prevent them from progressing to heart failure, which is much better than arriving a posteriori, where most of the irreversible effects of heart failure have happened. So a preventive study, if you will. That was quite risky for different reasons, because these, first of all, these patients are not easy to recruit. Second, because the ethics committee allowed us to use only the admitted dose for urological indication. I didn't tell you, but you may know that there is a high expression of beta-3 in the urinary bladder, and urologists use this drug to treat overactive bladder disease at 50 milligrams a day. And all the ethics committee in these centers allowed us to use the same dose, not more. They were afraid of side effects. And so we were stuck with that dose, but we went ahead with it. And our objective was to evaluate the effect of mirabegron over 12 months as an add-on to standard treatment to prevent the progression of cardiac hypertrophy and fibrosis, to try and see whether we could recapitulate what we had found in mice. So we had stage B patients, maximum New York Heart Association 2 stage, who were at risk of developing FPEF. And it's multicentric, and it's a bona fide RCT. Very well executed, despite the COVID. The COVID hit us in the middle of the study, but we went to the end and recruited 296 patients. What was the result? First of all, we looked at primary endpoints such as changes in LV mass index by cardiac MRI. So all these patients had 3 cardiac MRI, baseline, 6 months, 12 months. We looked at changes in E2E prime and index of diastolic function by echo, also at 0, 6 months, and 12 months. We had a number of secondary endpoints. We did late gadolinium enhancement, we looked at fibrosis by MRI, left atrial volume index, lots of biomarkers. We even did maximal exercise capacity, and we looked at safety. And there comes a lesson of humility, because the results are completely neutral. Nothing happened. So same left ventricular mass, same E2E prime, no change in peripheral markers of metabolism. There are a number of reasons for that, and one needs to learn from such an experience. First of all, we were stuck with 50 milligrams. And we know that the amount of expression of these beta-3 is much lower in the heart, human heart, than in the bladder. So what is effective in the bladder may not have been sufficient for the heart. And moreover, we purposely selected patients that were not very sick at all, maximum New York heart stage 2, with just left ventricular remodeling, that were very well treated with standard of care, including anti-hypertensive drugs that are known by themselves to be anti-hypertrophic. So there was very little room over 12 months to see a prominent effect of beta-3 on top of standard treatment. We did see a hint of a change, but it was not so-called statistically significant. And so we published a paper in a very good journal in JAMA Cardiology, but we need to retest the hypothesis. My interpretation of this is that we didn't test fully the hypothesis in humans. We need sicker patients, we need longer-term follow-up, we need higher doses of mirabegron or more potent agonists, such as vibegron. Actually, Henning Bungard, who published this study in heart failure patients, used 300 milligrams of mirabegron. I don't know how we obtained the approval of the ethics committee, but he used 6 times the dose that we used in our long-term study. So I leave you with this lesson of humility, hoping that you've accompanied me through all this journey on this particular aspect of our work. But I want to finish by acknowledging the important work of many people who accompanied me over the years on this and many other projects that I didn't mention here. Most prominently, Lauriane Michel, who is the postdoc who did most of the metabolic studies on the beta-3 mice. Nerea Hermida on fibrosis, Charlotte Farah on signaling, Ragus Fani, who did most of the mouse intervention studies, many others, including Alberto, who's in the room and collaborated with us for more than 6 months, and international collaborators, the name of whom you can read here. And I thank you for your attention.
Thank you, Professor Balligand. Grazie, Professor Balligand. Now the lecture magistralis is open for discussion. We start from the students, and then we move to the others. Question, curiosity.
Se volete potete
Anche in italiano,
fate pure.
anche se loro parlano meglio di me l'inglese. Bravi. Domande? Sì.
Allora gliela faccio in italiano. Intanto la ringrazio per la lezione, è molto interessante e affascinante. La mia domanda è, siccome questi recettori beta-3 sono espressi anche a livello, a livello vascolare, potrebbero avere un'implicazione nel trattamento dell'aterosclerosi?
So I'll reply in English if you don't mind.
Okay. Yeah. So since
I will
okay,
reply
okay,
in English.
great.
Yeah. So it's an excellent question. As
Thank you.
as you saw, we did find beta-3 receptors in the endothelium. And as we found in the cardiac myocytes, this beta-3 in the endothelium drives also an activation of nitric oxide synthase. And this promotes the relaxation. Actually, we found this with my associate Chantal Dessy many years ago, thanks to the help of another Italian company, Menarini,
Okay.
that was commercializing this famous beta blocker, nebivolol, because nebivolol is a beta-1 super selective blocker, but it also has agonist activity on beta-3.
Oh, I didn't know that.
And so,
Right.
yeah, that's why it's a vasodilator. It's very well known by clinicians. So we found, but obviously we went beyond nebivolol. We did find the expression of the beta-3 by immunohistochemistry. We confirmed this by RT-PCR. We confirmed this with pure beta-3 agonists. So you're right, there's beta-3 in the vasculature and it relaxes. And by producing NO, it's expected to be protective. We haven't gone all the way through redoing all the metabolic analysis, but it's a very interesting question because the endothelium has also a specific metabolism that may drive its phenotype, including through reactive oxygen species. Professor De Caterina is a world expert in, reactive oxygen species and atherosclerosis. So I would suspect that activation of this endothelial beta-3 would be protective, but we don't have definitive proof for that.
Thank you.
Quindi ringrazio l'allievo Luca Maria Francesco Tognari e ovviamente quando venite presentatevi, come facciamo sempre, oramai siamo oltre la 10ª edizione di Orizzonti e quindi si fa sempre. Allora, altre domande? Prego.
Good evening, Professor. My name is Giorgio Mais, and I'm attending the 3rd year of medical school. Thank you for your presentation. And I wanted to ask, since it has been described the role of this beta-3 adrenergic receptor in remodeling heart, whether if it was possible that it is also involved in the developing of the heart during the embryonic stages of its stage, and if it's possible that it can cause also some anomalies that have not yet been undisclosed.
It's an excellent question. I don't have data to answer your question because this would necessitate a longitudinal study of the expression of this receptor through different stages of development, and especially probably in cardiac progenitors, whatever they may be. It hasn't been done. It hasn't been done by ourselves, even though we have the tools to do that, but we haven't found the time to do it. It's a very good question. And so what you would expect from that, I can give you an indirect answer to your question, because even though we didn't confirm the expression of the beta-3 in progenitors, what we know, and we've published that, is that production of nitric oxide in some of these cardiac progenitors promotes the differentiation, at least in our in vitro model. And we published this in cardiovascular research years ago with cyclic GMP mediated through a modulation of the Wnt beta-catenin pathway, which is key for the transition from proliferation to differentiation. So I would suspect that if the beta-3 is there and drives production of nitric oxide, it would then do exactly what we found before, which is a transition from the proliferative state to the differentiation of these progenitors. But I don't have the data to answer you now.
Thank you.
Bene. Other question from the students? Prego.
Hi, I'm Sofia Maria Chavarella, and I attend the 1st year of the medical school. I was asking if the role of this receptor of beta-3 in regenerative medicine, so if there are some evidence studies.
So it's close to your colleague's question.
Yeah, I know.
And again, I don't have data to answer your very interesting question. But again, building on our previous paradigm, I would think that it may at some stage promote the differentiation of progenitors. But I have to tell you that even though there was a hype and a big excitement regarding the possibility of regenerating the heart from some specific progenitors, the enthusiasm is clearly dropped because there's no, first of all, these cells are very, very few. And there are very, very limited approaches that have shown, a true promotion or re-entry of these cells into the proliferation stage. I mean, Fabio has data with the Malrojac that is a proof to that, but whether there is any relationship between this phenomenon and the beta-3 is something that's completely speculative. So my answer, simple answer is I don't know, but it's a very good question.
Thanks.
Okay. Students? Yes?
Good evening. I am Matteo Venturi. I'm attending the second year of Merit School. First of all, thank you for the amazing presentation. I was wondering, since glucagon and adrenaline have always a synergic action, if also glucagon played an active role in cardioprotection.
I don't have, again, data to definitively answer your question, and I'm not aware of data that have specifically addressed your question. But maybe other specialists on metabolism may have more hints on this potential effect of glucagon on remodeling. Does anyone know of data on this? Well, if Fabio doesn't know, then probably nobody knows.
Okay, thank you anyway.
Good question. Students? Okay, now the session is open also for other students, even a bit older. Yes, Professor De Caterina, please.
Raffaele De Caterina, former student of the Sant'Anna School. And thank you, Jean-Luc, for this great overview of this lifelong path that you have gone through the characterization of this receptor. My question is related to the well-known favorable effects that beta blockers, classical beta blockers have, especially in heart failure. Both beta-1 selective and non-beta-1, so beta-1, beta-2 beta blockers. The question is, what is actually known about the effects of classical beta blockers on the beta-3 receptor? And could it be that by inhibiting the getting of the classical beta blockers on the beta-1 or the beta-2 receptor, you actually have a stimulation of the beta-3? And this is probably testable, and I'm sure you have done some panel searching on the relative activity or inhibiting activity of the classical beta blockers. So can it be one way by which the beneficial effect of classical beta blockers act?
This is logically deduced from the paradigm that I illustrated, and we wondered for a long time whether this was the case. And then we did exactly what you proposed. We looked back at the old pharmacology of all the beta blockers 1 and 2 that are used in the clinic. And surprisingly, nobody really did a thorough pharmacological analysis of the affinity or the potency of touching the beta-3 by these beta-1-2 blockers. So there is a complete lack of data. But actually, it's another Italian story, or partly Italian story. we developed a collaboration with Temple, and Fabio knows this university in the US, where a young scientist actually tested the effect of classic beta-1 blocker on cyclic G generation. And she found that beta-1 blockade actually would promote the NO cyclic G pathway, which is exactly what we would expect from a beta-3 activation. So if I remember well, at the time she used metoprolol. I'm not sure about this, but at least one of the classic ones. So this is the only piece of data I know that, that would go in line with your hypothesis that some of the benefit of beta-1 blockade would be through the recruitment of this NO cyclic G pathway.
Okay, I have a question. Did you detect some gender-based, sex-based, or age-based difference?
Not done. Not in mice. We exclusively use males because of logistic limitations. We should do, obviously, studies in females. In terms of human data, we obviously included females in our cohort of the beta-3 LVH study, but we didn't find any gender effect. But obviously, since we didn't find much of an effect overall, it's difficult to, but we could have expected, more prominent effect in the female sex, but we didn't find this. Unanswered question again, but important aspect that should be explored.
Okay. Yes.
Thank you for the wonderful talk. So I have a question that is related to the first part, the transcription factor analysis binding site. So when you did your analysis, you picked NRF2, but I saw that there are many interesting, actually, transcription factors. You got SRBF1, PPAR gamma, and also relay. So I was wondering if you have also analyze if some other, transcriptional pathways could be involved, like inflammatory pathways or those de novo lipogenesis pathways in the effect that you observed. And the second question is more related to the human.
Let me answer the first. So excellent question. And actually, the answer is part of the discussion in our paper that's coming out in circulation very soon. We looked just theoretically. So we did a literature search and we looked at all the downstream classical genes activated by these transcription factors, including SREBP, PPAR gamma, as you said. And there is good evidence that some of these, at least two, if I remember well, are involved in anti-hypertrophic effects. So they may well participate. But we haven't validated function. We focused on an RF2 because based on the phenotype that we had through unbiased approach, an RF2 was clicking all the items. Basically, all the genes were in its pathway. So we focused on that one, but you're right, we should go back and re-examine the others. And your second question?
Oh, so the second question is related to the clinical trial you performed. So you were saying that beta-3 is really important for white adipose tissues and those type of function. So I was wondering if instead of seeing an effect on the heart of your patients, you could see more a metabolic effect related also to, I don't know, activation of fatty acid oxidation, thermogenesis. Do you have some data related to this?
No, we don't have a full metabolic characterization in our beta-3 LVH cohort. We did some, there was a sub-study with PET. So, but again, very little activation by FDG with mirabegron, at least at the dose that we use and on the short and on the time frame that we used. But that's the only insight that I could give you. In terms of the other circulating biomarkers, we looked at classic lipids. We didn't do a full metabolomic analysis. We still have the samples, by the way. If you or someone else is interested, we could do a more thorough analysis. But at least in terms of triglycerides, lipids, we didn't see a prominent change.
Thank you.
Thank you.
We come back to students. Curiosity? No. I was watching your slides, and while you were showing this nice data,
What? I'm sorry.
nice data that you were showing, and I was thinking that is the pathway that you investigate is quite similar to the pathway activated by sulforaphane.
Mhm.
And you can find sulforaphane in cabbages.
Yeah. How do you say this in Italian?
Is sulforaphane is in cavolo.
Cavolo, sì.
Cavolo. Cavolo di Bruxelles too.
Sud Bruxelles. Esattamente.
Yes, right. Sprut Bruxelles.
Infatti, sì.
And so no, you know,
Sulforaphan is
yeah,
meant to post-translate
is the same pathway. Amazing. And we already published a paper on Journal of Translational Medicine where we demonstrated the anti-hypertrophic effect of extracellular vesicle released by fibroblasts pretreated by sulforaphane, that you have an anti-hypertrophic effect. And it is related to an increased uptake of this vesicle by cardiomyocyte instead of the other cells. Means that doesn't change the cargo, but change the uptake of the vesicle.
The destination of the
Right.
cargo.
So maybe in your case, you have maybe the receptor that is activated by sulforaphane and induce the release of extracellular vesicle could be the same like the beta-3, because the results are amazing.
Although, in my understanding, sulforaphane acts by, again, a covalent modification of proteins on tire residues again. So
Is dose dependent? It depends on the dose, because you have a hormetic effect, dose dependent. So it depends on the dose. But the pathway is the same. It's KEAP1, ARAE, and even related to MPK more than PKG. So
AMP kinase.
MP kinase.
It's fascinating because a few years ago, we published a paper showing that beta-3 activates AMP kinase.
So
all right,
just eat sprout or Brussels, and you have your favorite activity.
Thank you.
Yeah, you're welcome. So, okay, thanks.
Good evening and good to see you again today. And I would like to thank you for.
Please introduce yourself.
So I'm Jalil Daher, and I'm a visiting scientist coming from Lebanon, from University of Balligand. So I was saying good to see you again today, and I would like to thank you for this very interesting and captivating talk. So I have a small question regarding this beta-3 in endothelial cells in the context of endothelial cell dysfunction, because this is what I work on actually. And I would like to, and I know that you work with LDL also and oxidized LDL. So I would like to know whether there are some data at this level regarding the effect of oxidized LDL on the expression of this beta-3 in endothelial cells, because as you were saying, it has a bona fide beneficial effect in producing nitric oxide in endothelial cells, and this leads to vascular relaxation. And then the question is, because from our model, oxidized LDL model, we know that oxidized LDL also increases NRF2 expression. It is a transcription factor actually, which is induced by oxidative stress. So there are some kind of overlapping pathways, it seems, between the two. So yeah, basically, I would like to know more about this if you have some data at this level. Thank you.
So fascinating. I like your interpretation that there are crosstalk between different signaling pathways. So this is typically a model where systems biology might help. So in this case, we should collect, different data from different omic strata and try to put all this together and try and see how these interconnect with each other and where are the pathophysiologic nodes. I would suspect that, again, NRF2 would be one of the key nodes in the reconstituted systems biology pathway. But I don't have proof so far. What I can tell you is that in terms of LDL and endothelial function, as you know, because you work on this oxidized LDL or the pro-atherogenic, oxidized LDL obviously are oxidized through increase in reactive oxygen species. I would suspect that if beta-3 does in the endothelium what I showed it does in cardiac myocyte, the antioxidant effect would contribute to reduce the LDL ox and would protect from atherogenesis. It's pure speculation. I don't have data to answer that. But if you'd like, come to my lab and we can verify this.
Okay, thank you so much. Thank you.
You got it. Okay. Yes.
Jean-Luc, this was a great recapitulation of fantastic science over 30 years. I have scientific questions, but I'm not here to ask questions in this moment. There are many students here and trainees, right? Some of them possibly are interested in experimental research. In particular, I hope also in the field of cardiovascular experimental research, which is minoritary in our country and I would say, to some extent also in Europe, right? What would you recommend? What would you say? Any encouragement, any discouragement? So this is what...
Certainly not a discouragement. You shouldn't be, you shouldn't feel afraid from the lesson of humility that I showed you, with the neutral clinical trial. Clinical science is difficult, but it's doable if you have the right hypothesis, if you talk to the statisticians to design your study well in advance, if you have a very strict recruitment criteria, and if you really are persistent, as my collaborators were through the COVID period, we've reached our target of 297 patients. So my message is persistence. If you believe in an hypothesis, you should, put all your efforts until your curiosity is satiated. So be curious and have the courage to try and test your hypothesis. And persistence always pays.
Okay, we can say that with this message, we can say thank you to Professor Jean-Luc Balligand. E ringrazio anche gli studenti e tutti coloro che hanno partecipato oggi e a breve saprete quando e chi verrà a Orizzonti Medicina e Biologia 2024-2025. Grazie. Invito il professor Balligand, ora c'è la parte clou, della parte finale, che c'è tutto un... No, prego, venga qui. Gli allievi, ora lascio agli allievi, c'è tutta una fase finale del libro, una serie di doni, la spilla e l'attestato, quindi io me ne vado. Ragazzi, per cortesia.
Professore, noi la ringraziamo per essere stato qua. Noi consegniamo questo cioè il signature book per lasciare un pensierino agli allievi. E c'è una penna.
prestigiosi, io
In ordine cronologico.
in ordine tu mi dici dove filmare. Sì. Orizzonti. Ecco.
Ok, grazie mille. Poi abbiamo un attestato che rilasciamo sempre ai nostri ospiti, che è l'One of Us. Questo è per lei. Poi abbiamo altri gadget, abbiamo la spilletta e vi lascio tutti qua.
Grazie mille.
Facciamo una foto con l'One of Us.
Sì, se può aprire il...